Thank you, Koei. Thank you for nice introductions. So even though my talk title is focusing on some continuous glucose monitoring, I would like to show more results rather than focusing on some methodology and some analysis for the general audience. So here is the glucose patterns leading to type 2 diabetes. X-axis is age, Y-axis 2-hour glucose. The each dot stands for mean over 50, the PMA Indians. As you can see, glucose does not change much before reach the diabetes, which is the 200. So glucose does not increase much until reaching the threshold and sharply rise. So then we can think about the good biomarkers before reaching the threshold. So what are the good biomarkers? So robustness to detect the progressive disease, which means that that quantity already decreases before the onset of disease. Counterexample is glucose, as we see in the previous slide. Some prediction of the disease typically assesses log analysis and survival analysis. That's leading to good predictor. If possible, that the markers identifying the metabolic characteristics, so leading to personalized therapies. So today, let me show the result slide, then coming back. So this is the result that I want to show today. So x-axis is a noble mark. I'm going to explain today. The y-axis is mean glucose. So as we see, the glucose is slightly changed and shooting up. But as you can see this, the change of new biomarker dramatically decreases from, say, from the normal to the red one. But glucose is not increases much. Okay? Okay. So this is exactly symmetric at the PIMA data. So as you can see, this is symmetric. So this is today, I'm going to explain this. So now contents, for detailed contents for today's talk. The first, my talk composed of 4 parts. First one, summary of current metabolic surrogates during the various glucose challenge tests. Second one, noble mark during the standard oral glucose tolerance test. Third one, noble mark during the continuous glucose monitoring. The first one, clinical application. The first is personalized intervention. Second is detecting the subject high risk in the plane of disposition index. And third one, heterogeneity and homogeneity across different ethnic groups. Here's a gold standard measurement of metabolic parameters. The hyperinsulinemic and euglycemic clamp typically measure the peripheral insulin sensitivity. Hyper-glycemic clamps measure for beta cell functions. So insulin resistance, which is reciprocal of insulin sensitivity, and beta cell dysfunction are key pathophysiological factors for onset of type 2 diabetes. 2 clamp experiments are generally accepted as gold standard to measure the 2 risk factors, which are insulin resistance and beta cell dysfunction. Beta cell function relative to insulin sensitivity, we call disposition index. In this case, CDI, CLAMDI, insulin sensitivity times insulin secretion, is considered strongest metabolic predictor for T2D. However, the claim is not practical for large-scale study because it's required up to the 4 hours experimental time and also required highly skilled labor, and so this is not cost effective. So next, we're going to see the IBGT, the intravenous frequently sampled intravenous glucose tolerance test. So blood samples collected every 2 minutes to 30 minutes during the 2 hours study. They measured glucose and insulin. Beta cell function estimated by acute insulin response to glucose area under the curve insulin for the first 10 minutes. Insulin sensitivity estimated by fitting the minimum model to glucose. We call this the MINMA, developed by Richard Bergman, is a Banting medalist. Disposition index originally derived from intravenous glucose tolerance test. As you can see here, x-axis is SI, y-axis beta cell function AIRG. This curve, along this curve, the glucose is normal. So glucose is normal and constant. Say, just assume at the beginning, the SI is 2.0, AIR 400, they product 800. As the insulin sensitivity decreases by aging or obesity or something else, then as a beta cell, as an insulin sensitivity decreases, accordingly, beta cell function increases like this. Beta cell insulin sensitivity decreases up to the 0.4, and beta cell functions increases 2000. That product is the same. Their main normal glucose maintained. But DI decreases by decreases SI or decreases beta cell function or both. Then glucose increases, become high risk to develop diabetes. Now look at that current metabolic estimate during the OGTT, oral glucose tolerance test. So insulin resistance or insulin sensitivity estimated by HOMA-IRR. This one calculated by fasting insulin and fasting glucose. And quickly, also same, but with the log scale. Matchda index estimated by fasting glucose, the mean glucose, fasting insulin, and mean insulin during the OGTT. And beta cell function estimated by HOMA beta. Also, this is estimated by fasting insulin and fasting glucose. And insulin generation index, IGI, is calculated by increment in insulin between 0 and 30 minutes divided by glucose increment. Excuse me. So relative beta cell function and FDI, fasting disposition index. So this is the HOMA beta times reciprocal of beta cell HOMA IR. And well-known ODI, oral DI is defined by IGI times Matchda index. So all these ones are built in by algebraic formula, just using single or average measurement of glucose and insulin, rather than using glucose and insulin profile. So current mathematical model-derived estimates require frequently sampled OGTTs, and up to the 6 hours, at least 11 glucose and 11 insulin, 11 C-peptide, the Covallis model, Maris model. But they are not applicable for large-scale epidemiology studies. The standard OGTT, which means insulin and glucose collected every 30 minutes up to 2 hours, they underwent these really clinical settings. However, current mathematical models are not applicable to estimate the metabolic parameters with the standard OGTT. So my methodology focuses on standard OGTTs. Now, this is the novel metabolic parameters using during the standard OGTTs. So this model developed by ordinary differential equations. So, excuse me, the glucose concentration is balanced by glucose concentration balanced by glucose influx from 75 gram OGTT and endogenous glucose production from the liver, HGP stands for, and our glucose outflux, which is insulin-independent glucose uptake, the EG0, and insulin-dependent glucose uptake is the SI times I. The SI stands for insulin sensitivity. Also, insulin concentrations in the balance by insulin secretion from the pancreatic beta cell and clearance by typically happening in the liver. In here, we stand for sigma is the beta cell functions, secretory capacity of the beta cells. So we're going to estimate those insulin sensitivity SI and beta cell function sigma. So estimation SI sigma input data and glucose and insulin at 5 time points, 0, 30, 60, 90, 120, standard OGT. Then we're going to define the MDI. MDI stands for mathematical model derived disposition index, SI times the sigma. So I'm going to show the simulation for this. As you can see, the red one, glucose, and blue one, insulin. So the X marker is the data, the curve is the simulation. So just takes some 5 or 6 seconds. So for estimate SI and sigma for one patient, so they report, and the sigma is beta cell function, and SI the insulin sensitivity. Just take 5 to 2 seconds. So if you simulate, fit 100 patients, it takes less than 6 minutes. So very short. So we're going to estimate SI and sigma with standard OGTTs, which means glucose and insulin. But fortunately, we can estimate that MDI without insulin. So now MDI, mathematical DI, MDI with insulin, without insulin. So this is the MDI with insulin, the same as previous slide. Now we can estimate MDI without insulin. So we need only glucose at time 0, 30, 60, 90, 120 minutes. But we cannot estimate SI and sigma separately. However, we can estimate MDI. So we want to define that MDI without insulin. So let me show why this works. So actually this work awarded by Young Investor Award in ADA. So I'm going to show the rationale why we don't need insulin. So showing some mathematical skill, I'm going to just show with the picture. So this is blue dot, this initial gas. X-axis is SI, Y-axis is sigma, beta cell function, the actual DI plane. So the blue dot is initial gas. And green dot is SI and sigma without insulin. So along this curve, the glucose is constant, which means that kind of error between data and simulation is a minimized curve. So along this curve, the shortest distance is going to be this one, green one. So without insulin, estimate the point of this green one. Actually, the red one is with insulin. If you see the next slide, this one. Now we're going to use the insulin data, then the feeding identify red one is the right one. However, along this line, say highlighting the curve, that blue one and red one, they product at the same. So that's why MDI without insulin is possible. Actually, this is underlying background, mathematical background is kind of non-dimensionalization, kind of risky. So actually, our program director, Dr. Sujing Wang, she did some pioneer work, rescale in diabetic, mathematical model of diabetes. So now model validation. So I'm going to use the data from the collaborator from Pittsburgh, as Silva Selenian. So this is from the obese adolescents. Each patient did 3 experiments, 2-hour OGTT and 3-hour hyperinsulinemic euglycemic clamp and 2-hour hyperglycemic clamp. So the patient is the glucose tolerance is diverse, include NGT, IGT, type 2 diabetes. So now I just for the validation, and now x-axis is left panel insulin sensitivity with the model, y-axis is a clamp SR. So correlation is good. The right panel, the first base insulin secretion in y-axis, x-axis is a model drive beta cell function. So the correlation is really good. Also, second base is similar result. Now we compare the clam DI versus model DI. There are also strong correlation. Now we compare MDI with insulin without insulin. That correlation as 0.92 very strongly correlate, and also this slope is close to 1. So we can use the MDI without insulin rather than MDI. For the double check, I used a different data set from South Korea. So I compared MDI versus MDI without insulin. So as you can see, now R is 0.98, strongly correlate. Now we see that how that our MDI new markers predict the glucose, the mean glucose and glycemic excursion. So as you can see, I showed you previously, say MDI and mean glucose strongly correlate, but the decreases amount of at the beginning of diabetes that MDI dramatically decreases. However, the glucose slightly increases. So which means these are really good biomarkers. Also, the MDI predict glucose excursion very well. Now, really good predictor. So we compare using log analysis, we compare MDI and ODI to predict type 2 diabetes. So MDI is outperformed. And same for the pre-diabetes. But this one, cross-section, so far, data is cross-section. So we need longitudinal data for the real validation. So I took the longitudinal data from the Korean Genomic Epidemiology study from Dr. Sang-Soo Kim, Busan National University Hospital in South Korea. So at the baseline, 5,742 non-diabetes. The OGTT underwent every 2 years. The outcome is diabetes. This is 14 years longitudinal study. So as you can see, MDI without insulin outperform. Then the current diabetes criteria, so which are fasting glucose and 2-hour glucose, A1C, and ODI. So actually this one, fasting glucose is worst. Summary so far. So a model insulin sensitivity and beta cell function and DI agree well with the CLAM in obese use. And MDI without insulin correlate with MDI with insulin in obese adolescents in Korean data. MDI without insulin good and robust predictor. Now let's move to CGM. So now relative beta cell function during the continuous glucose monitoring. So continuous glucose monitoring, this is a real-time glucose monitoring. Typically, they control the daily average glucose and glycemic excursion. And glucose measurement collected in interstitial fluid. So I'll denote ISF glucose, and insulin measurement not collected. So this one is a less invasive and relatively cheap. So right panel, this is a typical glucose, blood glucose and ISF glucose pattern. As you can see, peaks of ISP glucose tend to delay and decreases. So question is, can we estimate relative beta functions with the CGM data? Yeah, current mathematical model cannot do this because they require insulin measurement. Our model can do this. As we can see that we can use our model. Now, goal is to estimate relative beta cell function, MDI, CGM, during the CGM. Now it's step 1. So the model, and we assume discrepancy between blood glucose, IS glucose uniform across patient. This is just for assume. And we need the data. The data set for step 1, the blood glucose and ISF glucose during the OGTT, which means patient during the OGTT where the CGM sense during the OGTT. And in this case, glucose load the same for everybody, same for all patients, this 75 grams. So this is kind of the mini project to the estimate relative beta cell function CGM. So in this case, so we're going to estimate this SI and sigma, but we're going to estimate SI sigma, the input data, ISF glucose at 5 data point, 0, 30, 60, 90, 120. So we're going to define MDI ISF equal, that's SI times sigma. So this is the result. This is the result, say, the left panel, the mean glucose, y-axis, and x-axis, MDI with ISF. So excellently correct, R is 0.96. And right panel, it's glucose excursion. So also strongly correlated. Now step 2, now relative balance function during the CGM in a free living conditions. So assumption same, discrepancy between blood glucose and ISF are uniform. Now we need the data. We need the CGM data under free living conditions for step 2. But the glucose load will be estimated with carbo intake. So basically glucose load is different for everybody. So we need input data here. So we're going to estimate SI sigma again, but input data, CGM data during the real meal, 25 time point. So basically CGM device collect the glucose every 10 minutes, every actually 2 minutes or 10 minutes. So we need the data every 10 minutes to up to the 4 hours. So, and also we need a total amount of carbo intake during the meal. So in this data, our collaborator estimated the carbo intake, say ChemPro 5.0, the web version. This is published by Korean Nutrition Society. With this, we're going to estimate N, sigma, and SI. And then output going to be estimation, SI, sigma, and glucose appearance rate. So we're going to use that SI sigma, MDI, CGM, SI times sigma. So I'm going to show a simulation for this. So this is a CGM feeding. Yeah, here. The red cross is CGM glucose, and red curve CGM, the simulation of CGM, and blue one insulin. So we don't care about insulin. It takes more time because this is 25 data point. So now it takes a little bit time, but now they want to report the SI and sigma value. Yes, a little bit takes time to this many data point. Yeah, now report here. So this value, SI sigma is 99 and SI 0.01. But based on my experience, 99 too big. This doesn't make any sense. However, their product really makes sense. Even though sigma balance function is very strong, but doesn't matter. Their product is really true value, but each individual sigma SI is not correct one. Okay. So this is the result. The mean CGM versus MDI with the CGM. So also MDI CGM in capture the mean CGM excellently. So I'm still doing some glucose excursion part with some compare the CGM parameters, say for example Maggie or some parameters still going on. So preliminary are really good, but I haven't finished yet. I'm not going to show for that part. But anyhow, that MDI CGM captured mean CGM glucose. Summary so far, mathematical model enabled to estimate relative basal function during the OGTT and CGM without insulin. MDI CGMs predict mean glucose. MDI without insulin, MDI and CGM as really cost effective because doesn't need the insulin and effective for large scale epidemiology study and also some potential beneficial for patient care. Now it's a really clinical applications. So this is a personalized intervention with estimate SI and basal function. This is same data from the Busan National University Hospital, South Korea. So as you can see, this is the insulin resistance group. The basal function is still intact. Now, this is right low corner. This is a weak basal function group. Insulin sensitivity is intact, but basal function really kind of deteriorate. So, as you will see, the black is the NGT, the blue is a pre-diabetic, excuse me, red is a type 2 diabetes. So model drive the basal function SI characterize glucose tolerance status. And also on top of that, insulin resistance is a manageable risk factors. Lifestyle intervention could be more effective with insulin resistant subject than weak basal function. So insulin sensitive, insulin resistance and weak basal function group, but unfortunately it has the same level of DI. So this is insulin resistance and basal function group has the same level of DI. So the question going to be, which group is high risk? This is kind of out of question in terms of MDI. So now, we're going to detect the subject in high risk in DI plane. So we're going to use a different, we need a little bit different method. So we use that machine learning algorithm. So we fit glucose and insulin. Glucose and insulin data. So we have a subtype of 6 subtype of non-diabetes. So using some machine learning algorithm. In the literature, typically they use only glucose profile to have some subtype of non-diabetes. But we use both glucose and insulin that we have a 6 subphenotype. For example, as you can see, class C and class E, their glucose profile are the same, but the insulin profile different. Then question is, which one? Which one is more risk to develop diabetes? This is same as before. Okay? E looks like insulin resistance, but C is the weak basal functions. Okay? So now we used longitudinal data from COGES, then fit sigma and SI plot at SI sigma plane, DI plane. As you can see, class C is the green one. And E is here, left upper corner. As you can see, right panel is survival analysis. So class C is kind of highest risk, followed by E, B, D, E, D, F, A. But in Korean population, A, B, C is 82%. So majority of A, B, C. So weakest basal function class C is highest risk. So now we're going to change the longitudinal change of cluster. So as you can see, A and B and B and C transitions are most frequently observed. As you can see, transition matrix say it is highest, A, B, and B, C. Therefore, A, B, C is the most common pathway to progression diabetes in Korean populations. So using just average, Korean population, as you can see, the arrow like this. Beta cell function does not come increase much, just kind of beta cell function decline as tiny decreases in insulin sensitivity. Now we see the Pima Indians here. So we don't have a much sample size. So I used linear mixed effect rather than subclass. So this in the individual, say the population curve is solid one, and dashed line individual curve. As you can see, population curve, beta cell function increases and decreases as insulin disease get worse. So totally different pathway between Koreans and Pima Indians. This is the heterogeneity between the ethnic group. Now look at this homogeneity of MDI. So now AUC glucose or mean glucose versus MDI without insulin curve is universal across different ethnic groups. So this is Japanese. X-axis MDI without insulin, Y-axis mean glucose or AUC glucose. This is Japanese. This is the African immigrant. They overlap. Regression curve almost same. And now added obese adolescent, same. Now Pima Indian, same. Now cystic fibrosis related diabetes, the same. So mean or AUC glucose versus MDI curve may be universal. So we really want to kind of find the cut point, optimal point in terms of MDI rather than glucose. So this data set are cross-section, so not meaningful to kind of optimal point. So we take advantage of longitudinal data set of Korean. So we found the cut point of MDI without to predict type 2 diabetes. So we found 1.94 is the optimal cut point in terms of MDI. To kind of support those ideas, I just plot MDI, x-axis, y-axis, AUC glucose or mean glucose. Excuse me. So two groups, red one progress group and black one non-progress group. So their baseline are different at MDI. And also different AUC glucose, but not that much. Excuse me. So we might guess between here some cut point or optimal point of MDI. Now we see 2-hour glucose and 1-hour glucose. Same pattern in 2-hour glucose. And their baseline MDI different, progress and non-progress. Same for 1-hour glucose, same for, excuse me, fasting glucose. But in here, fasting glucose at the baseline values of fasting glucose between two groups are not that different. They are not significantly different, which means fasting glucose is not good predict progression diabetes in Korean population. So summary overall, the model insulin sensitivity beta cell function DI agree well with conventional surrogate and outperform to predict dysglycemia. And model-derived insulin sensitivity and beta cell function could have potential to implement and personalized therapy. MDI without insulin MDI best predict diabetes compared to current diabetes criteria. Fasting glucose, 2-hour glucose, A1C, even ODI, based on longitudinal study. So combination of machine learning algorithm, model parameters identify subphenotype of non-diabetes, revealed most common pathway progression diabetes in cohort of Korean populations. This is potentially applied for other cohorts. This is conclusion. Excuse me. MDI is a really reliable indicator of dysglycemia, both pre-diabetes and diabetes. MDI without insulin is suitable for the large scale observation, intervention study, assess beta cell function. And sigma SI is a good surrogate for beta cell function and insulin sensitivity. Future direction is a longitudinal confirmation of MDI with CGM data with young African American in Howard University. So this is for your information. This I just summarized minimal data and the outcome. So during the standard OGTT, 5-glucose and 5-insulin that can estimate insulin sensitivity, SI, beta cell function, and also DI. Now, 5-glucose and plus 2 insulin point, and or 3-glucose and 3 insulin point, that also estimate SI, beta cell function, and disposition index. So I didn't show today, but strongly correlate. Excuse me. Now just 5-glucose, then we can estimate DI. Now 3-glucose, 0 minute, 6 minute, 120 glucose, and also can estimate the DI. Now CGM plus carbo intake, they can estimate the DI. And now finally, now fasting glucose and fasting insulin plus A1C. Then this one, we produce 6 phenotype of machine learning algorithm. This is still going on. So funding source, DK pilot study and brain pool program in South Korea and Howard University started fund. So here I'm going to introduce collaborator and data source, Sang-Soo Kim and South Korea, and Jimmy Kim, South Korea, Busan National University Hospital. And now is Clifton Bogaris. So he provided the Pima Indian data. And consultants in NIDDK, Ari Showman, and Stephanie, and Samnan. And collaborator and data source from Pittsburgh, Silva Selenian, Junior Kim. Thank you. So any questions, comments?
Okay, now is our Q&A session. Thank you, Joon. And then I saw there's a comment from the meeting chat from, I think it's Dr. Over Caprera said, congratulations to you about the nice work. Yeah.
Thank you.
You see that. And then, so during the Q&A session, welcome everybody. You're welcome to unmute yourself to ask question or type your question in the chat, and then I will repeat a question to let everybody know, and then Joon can answer. Okay. So there is one question in the chat. What are the clinical implications of the model-derived disposition index during CGM?
Yes, that's a really good question. So basically, as you see that glucose doesn't change much during the transition from NGT to prediabetes or prediabetes to diabetes. Doesn't much change. However, as you see that MDI CGM change much. Say, for example, my mean glucose during the CGM, say 5% increases. It might be okay, but CGM, MDI CGM, I guess more than 30 and 40% change. That kind of sign for the high risk. Is that enough for you?
Yes, I think it's answered the question. And then there comes the second question from the audience. Could you share us more about what is the next step of your project?
Yes, as I said in my future work, so we need kind of longitudinal confirmation MDI with the CGM rather than OGTT. So we need much data, longitudinal data with the CGM. So that's going to be, say, the future R1 and collecting CGM data, longitudinal CGM data.
John, great talk. Sorry, I was late. I missed the early part. I don't know if you touched on this or not. I was very impressed by the 6 subtypes in non-diabetes your model can tell. Do you have any data from diabetics, whether you have seen similar like heterogeneity with multiple subtypes? Can you comment on that?
Yeah, that's a very good question. Actually, our original work with some diabetic patient, include diabetic patient, but the result is very similar. But to predict diabetes, so I just get rid of diabetic patient and just to show non-diabetic. Yes, very similar result.
You also see multiples. Is it also 6 subtypes?
Yes,
Or-
single subtype. Yes.
Oh, I see.
Yeah.
But I mean, these 6 subtypes have different risks for diabetes.
Yes,
So I
different risk. So let me
assume
share
the-
screen.
Yeah, I was just
Yeah.
curious. The diabetics assume they come to diabetes through different pathways, right?
Yes.
Possibly. But after they arrive at diabetes, they still show 6 subtypes. That's very
Yes,
interesting.
They 6, I mean, they basically in this Korean group, they measure patient 82% population A, B, C. They change it from A. If say someone in Korea, they developed diabetes, they 82% probably they changes to from A, B, C.
I see.
Yeah.
Good. Wonderful. I have a second question, which is probably a bit bold. So I'm very, very impressed by the fact that you can get pretty good estimation of beta cell function from glucose tolerance test. My question is about the so-called beta cell function. It's what you are referring to, I assume, is a composite effect of the how healthy individual beta cells are, and also the total amount of functioning beta cells, namely beta cell mass, right? It's a composite effect. So were you able to distinguish the following 2 scenarios? Scenario 1, you have beta cells cross-board not functioning as well as normal. And scenario 2, you're losing beta cells, namely the beta cell mass is reducing, such as in type 1. Can you model tell the 2 scenarios apart? Or any indices can
Okay, so
give
that's
us...
really, really excellent question. So the short answer, with the current estimating beta cell function and insulin sensitivity algorithm cannot capture beta cell mass and beta cell function. Actually, you can see in the model, mathematical model, see this one. So here, beta and sigma, they product. So amount of insulin, secreted insulin cannot kind of dissociate beta cell mass and function. So current estimate beta cell function is not, but our original model, since 2016 and the chronology 2020, that we have this model, this has a more detail in beta cell mass and beta cell functions. So
excellent.
Yeah, there might be some potential, but yeah, I'm not sure that we need more measurement, like a C-peptide and some, but a C-peptide and also some other, insulin, some others quantity. Then we actually, we can discuss about this because a little bit this is a private, So I'm working with this part. So
excellent, yes, that has been a challenge, I guess, for the field.
Thank you for really excellent questions.
Thank you.
Okay, thank you, Sujin. And then there is one question from Emily in the chat. You showed a slide with glucose arrow and a 3-point, red, blue, and green. Could you explain more how you were able to get from your initial guess to the blue point?
Okay, thank you. Thank you for the question. So here, okay, so as you see, blue one, initial guess of SI and sigma. Now, as you can see, I'm highlighting curve. Along this curve, glucose is constant or error between data and simulation, glucose and insulin, it's just the glucose are the same. So from blue to green, this is shortest distance. So the fitting algorithm captured this green one is the right kind minimum error. So this is with the estimate with only glucose. Without insulin, only glucose data, that green one estimate optimal SI and sigma, but wrong one. SI, sigma is not the right one, wrong one. Red one is the right one. Now we're going to use insulin data. On top of that, insulin data, then captured. As you can see, that red one. Red one, this really highlight only 1 point. So SI without insulin, our feeding algorithm captured green one, with the insulin captured the blue one. However, their product, as you see, their product along this curve, highlighted curve, their product are the same. So we can use the MDI without insulin to without insulin as it look like MDI. That's the reality. So this is kind of graphic explanation, but as I emphasize that this is, I can explain this rescale or non-dimensionalizations. As our program director, Sujin Wang, she did in pioneer work in diabetic model in rescaling part.